Thank you very much, and thank you very much for everybody who remained on the line. So I'm a research staff member at IBM Research in Daresbury in the northwest of England. And the title of my talk today is the role of chemical heterogeneity in surfactant absorption at solid-liquid interfaces. And before I dive in, I just want to let you know that all the work which you're about to see was performed as part of a collaboration between IBM Research and the Science Technology Facilities Council. And this collaboration was funded by the Department of Business, Energy and Industrial Strategy. So the first question is, why are we interested in this topic? Well, the motivation is because the absorption of surfactants onto solid surfaces is ubiquitous not only in nature, but also in many industrial formulations. Indeed, it's central to the performance and function of many industrial products, such as cleaning products and detergents, to coatings, to lubricants, and other products. However, conversely, it can also impede the efficiency and performance of some industrial processes, such as enhanced oil recovery. And one of the biggest problems which one encounters when one wishes to investigate the absorption of surfactants onto solid surfaces is the fact that surfaces are almost always rough or display some degree of chemical heterogeneity or disorder. And we've seen in the literature examples how these factors can lead to variations in the number of absorbed surfactants, in the heat of absorption, variations in absorption isotherms, as well as differences in the shapes and structures of the absorbed micelles themselves. And we need to get a better understanding of this in order so we can produce better performing products for the applications which I've just mentioned. So in this work, we interrogated the global interplay between cohesive micelle forces, cooperative effects, and surface interactions for 4 model non-ionic surfactants absorbing on a solid slab. And from this exercise, we derived and demonstrated a computational framework for the design of nanopattern surfaces for optimal molecular absorption. So we employed a coarse-grained molecular dynamics simulation methodology called dissipated particle dynamics in this work. So this allowed us to access length scales and time scales large enough to capture the physics of surfactant assembly, as well as conserving the chemical specificity of the individual molecules and their functional groups. So here we have the 4 model surfactants which we used. These non-ionic surfactants composed of 2 DPD types. So we have hydrophobic tail beads, which were parameterized as alkane beads. And then we have the hydrophilic tail beads, or sorry, head beads, which are parameterized as polyethylene oxide functional groups. And by selecting these 4 model surfactants, we can interrogate the effect of varying the hydrophobic and hydrophilic ratio, as well as the length of the molecule and how this affects their absorption characteristics. So next we have the surface which we use in these simulations. So this was a cubic lattice of frozen DPD beads that interact with all the other DPD beads in the simulation. However, they are frozen in their position. So at the bottom layer of this 3-layer thick lattice, we have an impermeable layer, which is strongly repulsive and whose function is to prevent the permeation of molecules of water in the bulk from entering into the solid slab. In the middle layer, we have a nanopattern surface with or a pattern with hydrophilic and hydrophobic beads. And in the top layer, we have the same pattern replicated as we have in the middle layer. And the reason why we chose to have 3 layers was we found that this to be the minimum number of layers which is required in order to ensure that we have an impermeable solid slab. And this is due to the fact that we have in DPD the use of soft core potentials, which can lead to problems of permeability. So next we derived a surface generation algorithm, and by applying stochastic Monte Carlo approach using 3 input parameters, we can produce a range of bespoke hydrophilic, hydrophobic surface patterns. So of the 2 input parameters, of the 3 input parameters, we have 2 independent generating probabilities, Px and Py, and they describe the level of disorder that we have in the x and y axis. And finally, we have S tar, and this is the global target hydrophilic, hydrophobic surface fraction. So basically, if we want a surface to be 80% hydrophilic or 20% hydrophobic, then that is we have a 0.8 for S tar. So we generated 3 classes of surfaces in this work. So first of all, we had the purely hydrophobic and purely hydrophilic surfaces. And next we generated 10 disordered surfaces using different pairs of Px and Py, where the target surface fraction was 50% hydrophilic and 50% hydrophobic. And finally, we generated 8 striped surfaces where we have parallel hydrophobic stripes separated by repulsive hydrophilic regions. And we just have variations in the separation between the stripes. So we imposed a heuristic cutoff of 1.3125 in DPD length units to describe whether or not a molecule is absorbed to the surface or not. And we classify it as being absorbed if one or more of its constituent DPD beads was within this cutoff radius or length scale. And this is very much a heuristic length scale. However, the qualitative results in this work were unchanged by variations in this parameter. And when we performed absorption isotherms for the purely hydrophilic and purely hydrophobic surfaces, we can derive a baseline theoretical prediction, shown in this equation here, for what we believe the absorption isotherm should be for disordered heterogeneous surfaces with hydrophilic and hydrophobic components, which is weighted by the relative proportion of the surface that is respectively hydrophilic and hydrophobic. So here we have the absorption isotherms for the predominantly hydrophilic surfactants. So here we can see that the number of absorbed molecules per surface area increases with concentration, as one might expect. And this is shown by the discrete data points in the plots. And what we found was the absorption profile of these data points follows a Langmuir-like isotherm, which we know describes the absorption of individual non-ionic surfactants onto a surface, as one might expect. So we're able to fit for the purely hydrophilic and purely hydrophobic surfaces this Langmuir type isotherm with fitting parameters capital gamma and capital K. And that's why we have the solid curves at the top and bottom of each of the plots. And then we can apply our theoretical baseline prediction for the absorption isotherm for heterogeneous surfaces, shown by the curved line, the dashed line in these plots. But what we can notice, especially for the longer H6T2 molecule, is that there's a clear deviation between the simulated data points and the dashed prediction. And this is a result of enhanced absorption due to the presence of patterns on the surface. So here in the left-hand side, we have the percentage enhanced absorption for all the 10 disordered surfaces. And we can see it's especially prevalent or pronounced for the longer H6T2 surfactant. And on the right-hand side, we can see the spatial distribution of the absorbed molecules. And we can see that, as one might expect, they predominantly absorb onto the attractive hydrophobic regions. However, we do see in some cases that the absorbed molecules can protrude sterically across the border between, or boundary between, the hydrophilic and hydrophobic regions, and that may explain why we have slightly more enhanced absorption for the longer H6T2 surfactants. And for the predominantly hydrophobic surfactants, we have a different mode of absorption. So first of all, cooperative effects dominate. We have the formation of micelles in the bulk, and therefore we have quite slow dynamics with the assembly, diffusion and dissolution of individual micelles. But also the absorption process itself is significantly different. So for the predominantly hydrophilic surfactants, they absorb pretty much independently of one another onto the surface, whereas for the predominantly hydrophobic surfactants, we have a collision of individual micelles with the surface, upon which they restructure into more energetically favorable configuration. So from that point of view, there is significant sampling difficulties in these simulations, and therefore we expect reasonably large error bars for the figures shown shown in this slide for the hydrophobic surfactants. Nevertheless, we can see qualitatively, again, we see significantly enhanced absorption, particularly for the longer H2T6 surfactant. And we also see the presence of bridging, which is where an absorbed micelle can bridge or span across from one hydrophobic attractive domain across a hydrophilic domain to another attractive hydrophobic domain. And we can see this in greater clarity, or greater clarity of this phenomenon in the following simulations when we have the two parallel hydrophobic stripes. So on the left-hand side, when we have very small separations between the stripes, we have a bridging structure, which is, I can see, a large sort of hemi-cylindrical type structure. And the very right-hand side, we have stripes which are very far apart, they're disconnected and independent of one another, and we have hemi-cylindrical type structures. But in between, we have this competition between the attractive cohesive interactions of the micelle structures themselves and the repulsive surface interactions of the repulsive domain in between the stripes. And this leads to a sort of rich morphology or of different flat and curved bilayer structures. And another curious thing we notice is, well, when we have the same surface, or the disordered surface, but for different simulations with different molecules in the bulk, we found that the regions where the molecules are more or less likely to absorb to be relatively the same, as we can see in this plot. So in order to interrogate this, we developed something called a bare surface potential from the bare surface pattern itself. And we applied a Gaussian blur analysis to this bare surface potential, which has one tunable parameter, namely the standard deviation sigma. And we fitted this to the spatial distribution of the absorbed molecules themselves. And by doing this, we can tell that if sigma is quite a small number, then we have a very short-ranged overall effective potential. However, if it's quite large, we have a relatively long-ranged effective surface potential. And we applied this to the stripe simulations. We see that for the larger absorbed structures and for the cases where we see bridging, we generally have larger values of sigma and therefore more long-range effective surface potential. However, if we look at the disordered surfaces, we see that for the hydrophilic surfactants, we have more short-range effective surface potential relative to the hydrophobic ones. Similarly, for longer surfactants, such as H6T2, we have more long-ranged effective surface potential relative to H3T1. And if we increase the spatial order, we see reduction in the fitted values of sigma, denoting more short-range effective surface potential. So examining the results, we thought of an interesting thought experiment. So suppose you had a case where you wanted to maximize the number of absorbed molecules per surface area on a surface, but you have fixed surface composition. And that translates into a task where you have to optimize effective surface potential landscape. And what we can see here is that if your attractive and repulsive domains are too small, then you end up with shallow attractive wells from which surfactants can quickly detach. However, if the attractive domains are quite large, then the overall surface coverage or absorption would be limited to a few attractive domains that you have. So there's like a Goldilocks region in between these two extremes where you have a maximum number of absorbed molecules per surface area. So to conclude, what we found is that the number of absorbed molecules or overall global absorption was maximized for heterogeneous surfaces relative to homogeneous ones. If one increases the length of your molecule and its hydrophobicity, you get an increase in cooperative effects and absorption. Another interesting feature is that the characteristic lengths of these bridging structures exceed the length of the individual surfactants themselves. And this opens up the possibility where one can tune the surface patterning in order to calibrate the absorption characteristics both locally and globally. So finally, I'd like to thank my team who worked on this, especially two of my former interns, Jason and Sophie. So this paper is on the archive and just recently got accepted for publication by the Journal of Chemical Theory and Computation. So hopefully I'll be out soon. So thank you very much for your time, and I look forward to any questions which you may have.